Good. My name is Lynette Corona. I am the Healthcare Program Specialist at CDH's HI program. I will be for today's webinar. Today's webinar is entitled The Mechanisms of Resistance in CRE in California. Our Dr. Niaz Banai and Sam Borla. We'll wait at the end of each presentation to address any questions to the presenters. Please questions by using the chat or Q&A panels, and we'll make sure to get them answered. And I will now turn the presentation over to Dr. Erin Epson, the Assistant Chief and Public Health Medical Officer with HA program at CDPH.
Good morning, welcome, and thank you all for joining us. Today's was part of our California Antimicrobial Resistance Laboratory Network, where our goals are to enhance situational awareness of healthcare-associated antimicrobial resistant pathogens and to connect healthcare facilities and laboratories to additional laboratory testing resources to support care and infection control activities. We have enough time for our presenters today, but I do want to bring up one upcoming project we're working on as part of our network, and this is to assist clinical laboratories that are still using old breakpoints to determine carbapenem resistance in Enterobacteriaceae. This graph shows the number of acute care hospitals in California that reported in their National Healthcare Safety Network, or HSN, annual hospital survey that their clinical laboratory is using the updated breakpoint. And as you see, although we've made progress in the recent years, we do estimate that as of 2017, about 13% of California hospitals are still relying on laboratories that use old breakpoints for carbapenems. So if is not using the most updated breakpoints to identify carbapenem resistance in Enterobacteriaceae, please consider working with us in the HI program to update your interpretive criteria. In the near future, we plan to host an introductory webinar, which will feature laboratory experts in the field, as well as other hospital labs that have successfully gone through this project. We want to work individually with interested labs to discuss the process and develop plans for updating their breakpoints. Some issues we'll address include accessing the CDC AI isolate bank, going through the cation process of antimicrobial susceptibility testing, resolve discrepancies, and writing up a verification report. So again, please get in touch with us if this is of interest to you. Okay, objectives for today's webinar presentations are to describe and compare phenotypic and genotypic characteristics of CRE identified at a Silicon healthcare facility as well as in Los Angeles County. We characterize the prevalence and distribution of carbapenem-producing-CRE in California, and we'll discuss the public health significance as well as clinical implications of these diverse mechanisms of resistance among CRE in California, and then review the roles of public health laboratories in identifying carbapenemase-producing CRE. Today to be joined by Dr. Niaz Banai and Sam Borla. Dr. Banai is an associate professor of pathology and medicine at Stanford University School of Medicine. He's the medical director of Stanford Healthcare Clinical Microbiology Laboratory, director of the Stanford Clinical Microbiology Fellowship, and associate program director Pathology Residency Training Program. Is the antimicrobial resistance epidemiologist at the Acute Communicable Diseases Control Unit at the Los Angeles County Department of Public Health. So up we have Dr. Banai's presentation. As Linda mentioned, please type any questions you have in the chat function, and we'll make sure to get them answered. So without ado, I'm pleased to introduce Dr. Banai.
Thank you. Can you hear me?
Yeah, I hear you well. Lynette is going to tee up your presentation. Thanks.
Lynette, okay, so morning. Thank you for the introduction. It's a pleasure to share with your experience on what we have learned about the diversity of mechanisms in our carbapenemase-resistant Enterobacteriaceae at our health system here at the wanted to, as an introductory sort of slide, what I wanted to share with you is the timeline of microbial arms race. And what the slide shows is that every time we have taken a natural product and turned it into antibiotics and introduced it into patient care, shortly after, the organisms have acquired a genetic mechanism to detoxify these antibiotics. And
I'll interrupt. This is Sam. Could you actually share your screen? We're having trouble seeing your patient.
No worries.
That to make sure that that's possible.
So, tab.
Lynette, anything you need me to do on this end?
Now, at this point, if you can hit the Start tab and share desktop from there.
Start tab.
Left.
I see it, yes.
There you are.
Perfect. Thanks. Sorry about that.
No problem. So yes, there we are. So every time you have introduced a drug in patient care, shortly after, the organisms acquired a genetic mechanism to detoxify the drug. And the latest chapter of this microbial arms race is written by carbapenem-resistant Enterobacteriaceae that use carbapenemases and other mechanisms to detoxify carbapenems. So these bugs are considered superbugs, and they pose a serious public health threat. Just to quickly illustrate the point to you, I want to show you a case that we saw here a few years ago. This was a man that came to visit his daughter from the Indian subcontinent. His daughter is an engineer in Silicon Valley, and he had a urinary tract infection, and you can see we recovered over 100,000 of E. coli. And when we tested his organism with YTEC2, essentially almost every drug that we tested was resistant. And we had to go to drugs like fosfomycin and colistin to come up with something that we could potentially treat him with. So what I have to share with you today is our local experience in terms of telling you about the rate that we see at Stanford and the mechanisms that are responsible for our CREs, and what the in vitro susceptibility of our CREs to newer beta-lactam, beta-lactamase inhibitors. And then lastly, whether we have any transmission going on in our health system. And if time permits, I also will share with you what might be a future challenge with the way we are testing and predicting susceptibility for CREs. So the definition that is currently used to define carbapenem-resistant Enterobacteriaceae is a post-2015 CDC definition that defines Enterobacteriaceae as resistant to any of the carbapenems, including ertapenem. And the mechanism of resistance in these CREs is twofold, and they're not mutually exclusive. So the mechanism that's feared is when these organisms pick up a plasmid that encodes a carbapenemase. The carbapenemase is produced, translocated to the periplasmic space where it intercepts incoming carbapenems and detoxifies them or hydrolyzes them. This mechanism is feared because the plasmids also encode other mechanisms of resistance, and also this mechanism is easily transmitted nosocomially between patients. Second mechanism is inactivation of outer membrane proteins known as porins. In E. coli is outer membrane protein C and F, and then their analogs in other members of Enterobacteriaceae. When these are inactivated, the carbapenems cannot get in, and this always almost happens together with overexpression of cephalosporinases such as AMC and ESBL. And although these enzymes don't neutralize carbapenems, they're essential for creating resistance to carbapenems together with porin inactivation. So over the past 4 decades, about a dozen or so carbapenemases have been identified in various gram-negative odds. For the purpose of today's discussion, I want to focus on the 4 that are shown in red. These are the ones that are being transmitted globally. And if you look on the right-hand side, each one of them has emerged in a different country, and from there it has spread internationally. So imipenemase initially discovered in Japan and has since spread globally to Taiwan and the rest of the world. Verona integron encoded metallo-beta-lactamase was initially discovered in Verona, Italy, and then it spread to Greece, and from there also to the rest of the world. OXA48 was discovered in Turkey, and since then it has become endemic in Western Europe. KPC, Klebsiella pneumonia carbapenemase, is actually homebrewed. It was identified in North Carolina. From there, it spread to Northeast and also internationally to Colombia, Israel, and also China. And the most recent discovery was on New Delhi metallo-beta-lactamase, as its name implies, was endemic in India and Pakistan and Bangladesh, and from there it has spread to elsewhere. And you can see each of these 5 plasmid-encoded carbapenemase belong to a different molecular class of beta-lactamases, and here they're shown. So KPC belongs to molecular class A, OXA48 belongs to molecular class B, and the other 3 belong to molecular class B, which are metallo-beta-lactamases. So about 10 years ago, you didn't really have to know the molecular class that these enzymes belong to, but today it actually matters because molecular class predicts susceptibility to the newer beta-lactamase inhibitors. So whereas class A and D, so KPC and OXA48, are serine beta-lactamases and use their serine residue to attack the beta-lactam ring and hydrolyze it, metallo-beta-lactamases use their zinc to activate a water molecule to hydrolyze the beta-lactam ring. And based on the molecular class, you can now actually predict which of the newer beta-lactamase inhibitor combinations is going to work in vitro. So, for example, avibactam is able to inhibit class A and class D, so KPC and OXA48. So the combination of ceftazidime and avibactam will in vitro give you susceptibility against those 2 mechanisms, whereas metallo-beta-lactamases are not inhibited by avibactam. However, aztreonam is not hydrolyzed by metallo-beta-lactamases, so combine aztreonam with avibactam, which prevents hydrolysis of aztreonam by cephalosporinases like AMC and ESBL. You can actually get aztreonam combined with avibactam to give you activity in vitro against metallos. So going to our local experience, the first question that we wanted to answer is, what is the rate of carbapenem-resistant Enterobacteriaceae at Stanford Healthcare? So we looked over a 4-year period from 2013 to 2016, and out of 19,171 unique Enterobacteriaceae isolates, we had 62 CRE isolates from 61 patients, giving a rate of 0.3%. I should review that again. This rate is based on the pre-2015 C definition. If we include the post-2015 definition, the rate would definitely be higher than 0.3%. Even when you compare our rate to other institutions using the same definition, our rate is quite low. So when you compare us to national rates, Stanford rate is about 4 to 13-fold lower. So the national rate was 4.2% per 2011 NHSN data and 1.4% per 2010 Surveillance Network USA data. Our rate is also less than half of that reported by UCLA Health System, where they found 0.73% rate. So as you note here, both us and UCLA, so you can say California seems to have a lower rate than national rate, and that seems to be true. What seems to have lower rates. This is a study that was conducted in 7 communities in the U.S., essentially 7 states, and one of the findings is that western states have lower rates compared to eastern states. So Colorado, Mexico, and Oregon have lower rates compared to Georgia, Maryland, and New York. So, mission is our in the carbapenemase and Enterobacteriaceae that we saw during our study, what was the mechanism that they encoded? And so again, just to remind you, there was two mechanisms. We first wanted to find out whether our isolates had a carbapenemase. And so we went to this list of known carbapenemases, and most of them are plasmid encoded, but some of them are also chromosomally encoded, such as the serratia marcescens SME. And so we developed, used lab-developed tests at Stanford, and also in collaboration with Cepheid, we used their cartridge and also used the checkpoint assay, generally available, and we looked for existing carbapenemases, and also we looked to see whether our isolates encoded ESBLs and plasmid encoded AMPCs. And here's our finding. What we find is that out of those 62 CREs, about 9% of them have one of the 5 plasmid encoded carbapenemases and also serratia marcescens SME, and 61% of our CREs did not have a carbapenemase gene. I hope you appreciate from this graph that this is a very cool graph, and if you ignore the orange, orange that don't have a carbapenemase gene, you can appreciate that there's many mechanisms that we are seeing here. No single carbapenemase is predominating. In fact, the rates with KPC, OXO48, and SME were very similar. And most other institutions in the U.S., you don't get such a colorful graph. Instead, one mechanism predominates, and it is typically KPC that predominates. And so in that regard, we are very unique. And I can't explain it to you why that is, but I think it has to do with the fact that we are in Silicon Valley, and people are coming from essentially all over the world, and they're bringing their CREs with them, or carbapemases with them. And also another reason, as you'll see later, is that these are sporadic cases. We don't have transmission going on, so no one mechanism has established itself in our healthcare system. And probably also helps that we don't have long-term care facility where CREs become endemic. And so just going back to the study that I already showed you, out of 188 CREs that they tested, 90 of them were carbapenemase producing, and percent of them were KPC. To drive in the point that it is really unique that we don't see carbapenemase KPC predomination in our institution. So now that we have identified the mechanism of resistance in those that have a carbapenemase gene, we would be interested to figure out what's going on in these organisms that don't have a carbapenemase gene. So there are some phenotypic differences between the carbapenemase gene positive and negative isolates. So, for example, the ones that encode a carbapenemase have a higher MIC against imipenem and meropenem, compared to the ones that don't encode a carbapenemase gene. They have a lower MIC to imipenem and meropenem, and this has been illustrated before in other studies. So we think that the mechanism of resistance in the isolates that don't have a carbapenemase gene is porin inactivation, together with overexpression of cellsporinases such as AMPC and ESBL. And in fact, majority of these organisms that don't have a carbapenemase gene encode a plasmid encoded, majority of them are space organisms, which have chromosomal AMC, versus the organisms that encode a carbapenemase gene, minority of them are these organisms. But actually wanted to make sure that we were not missing any carbapenemases that had not been discovered yet in the ones that didn't have a carbapenemase gene. So we did a couple of functional assays to show to prove that when they don't encode carbapenemase gene, they also don't have carbapenemase activity. So we used two assays. The first assay is the carbapenem inactivation method, where you take your test organism, make a suspension with it, and drop a carbapenemase disc in there, and you hours, and then you take out your disc and you place on top of a lawn of reporter E. coli that are susceptible to carbapenems. And so if your isolate had a carbapenemase, it would have hydrolyzed all the carbapenem in the disc, and so when you place it on E. coli, the E. coli grow right up to it and indicates carbapenemase activity. But if your isolate didn't have a carbapenemase, then all the drug would be preserved in the disc, and you get a zone of inhibition. So using this method, according to the CLSI interpretive criteria, we could identify those that were carbapenemase producing and those that were not, and the indeterminate results were resolved with a multi-tough assay where you incubate your organisms with impenem as a parent drug, and if it's hydrolyzed, then it disappears and the metabolite appears, and you actually use a ratio to determine which is positive or not. So using the combination of the SIM assay and the multi-tough, what we found is that while 100% of the carbapenemase gene positive CREs were carbapenemase producing, 4% of the 38 CREs that didn't have a carbapenemase gene were carbapenemase producing. So it confirmed for us that, in fact, we didn't miss any carbapenemases in this group when we tested genetically. They just don't have any activity. They don't have a carbapenemase, which further points to the fact that the mechanism in these is protein activation together with cephalosporinases. So to further prove this, we worked with collaborators at Bruker and also UC Davis, where we developed a pipeline to take outer membrane proteins from our CREs, digest them, and we did tandem mass spec. And at the end, we get a spectrum. From here, you can actually not only quantify but also identify fragments that correspond to the outer membrane protein C and F of E. coli, and also the analogs in all the other Enterobacteriaceae species that we had amongst our CREs. So with this approach, what we could show is that when our isolates don't have a carbapenemase gene, 92% of them had lower expression of membrane protein C or F, and interestingly, similar percentage of the carbapenemase gene positive CRE, also showing down relation or inactivation of porins. So for most part, we can show that our carbapenemase negative CREs have a porin inactivation, and what's more interesting also is that having a carbapenemase gene is not enough. They combine that with porin inactivation, and this is consistent with the fact that if you just transfer a carbapenemase plasmid into a susceptible E. coli, the IC is not that high, actually, for carbapenems. So you really need to combine it with porin inactivation to get the high MIs that these clinical isolates typically have. So moving on to, now that we know the molecular mechanism, can we actually predict susceptibility to the newer beta-lactamase inhibitors? And so to remind you again, avibactam and laborabactam, which are FDA cleared, are able to inhibit Class A or KPC, and Class D corresponds to also 48. Relabactam is in trials, and it can inhibit Class A or KPC, but not others. And so the question is, can we accurately predict susceptibility of our isolates in vitro with these drugs combined with different lactams? And the answer is yes. We get exactly as what you would predict. So when you take meropenem, laborabactam, and ceftazidime, avibactam, essentially with avibactam, ceftazidime, avibactam, 100% of our carbapenemase gene negative CREs are susceptible. For most part, this is also true for meropenem and laborabactam. But for carbapenemase gene positive CREs, it totally depends on the mechanism of resistance. KC, OXA48, and SME are susceptible predictably. The metabelactamases are either resistant or intermediate. And you had a very similar finding with imipenem relabactam. Again, most of the carbapenemase gene negative CREs are susceptible. with the carbapenemase gene positive, it is only the KPC that's predictably susceptible. The rest of them are not usually predictive. We also tested ceftolozane and tazobactam just because it is not a drug for CREs, but we wanted to see what we find. And what's interesting is that about a third of the carbapenemase gene negative CREs or porin inactivation CREs are still susceptible, and the only carbapenemase gene positive CREs that are susceptible are the ones that encode an SME. As you remember, SMEs are susceptible to cephalosporins, and so ceftolozane is a cephalosporin, which makes sense. All right, so the last aspect of our study was to determine what is the nature of transmission of CREs at our institution. To be honest with you, I never suspected we had any transmission because look at the colors here. We are getting such sporadic detection with these CREs that producing carbapenemases that unlikely that we don't have one mechanism predominating, so it doesn't look like we have transmission. However, you can ask, was there transmission of these non-carbapenemase producing CREs? So to do this transmission investigation, we collaborated with Ami Bott and our laboratory members at Stanford, where we sequenced the genome sequencing on the 62 isolates and looked at the full genome or the core genome and determined whether there was any kind of genetic link. And the answer is no. No two isolates are identical. Our CREs, the closest that we came were in these three cleft pneumos, and one of them had no temporal association. The other two, CRE 24 and 25, were isolated 3 days apart from two different patients on two different wards. But the isolates are 10 single nucleotide variants different from each other. And so what it suggests to us is that they're not genetically identical, but perhaps they share a common link, and somewhere in their past they came from the same descendant. And so to summarize we found at our institution we have low CRE rate, and by doing comprehensive phenotypic and genotypic characterization, we identified sporadic occurrence of plasmid-encoded CREs, carbapenemase-producing CREs, and we did not see predomination by KPC like most other institutions in the U.S. We could predict susceptibility to newer beta-lactamase inhibitor combinations based on the mechanism of resistance that we detected, and for the healthcare institutions in California or North California, perhaps I should say, they can say that on-demand testing was sufficient for detection of carbapenemase-producing CRE and predicting their susceptibility to the newer beta-lactamase inhibitors. So I promise you that I would leave you with one more topic, and that's future challenges. And I want to show you a case that we currently have at Stanford that illustrates what the challenges that we might be facing in the future. And this is a 61-year-old liver transplant and kidney transplant patient who had respiratory cultures in April, and then in May had urine cultures. And you can see in April, the respiratory culture was positive for Enterobacteriaceae complex and pretty resistant to the drugs we tested, susceptible to aminoglycosides and Bactrim. And when we did genotypic testing, it was positive for KPC. And at our institution, we then we know that with K should be susceptible to ceftazine-avibactam, and in fact, it was at 1 microgram per mil. But then fast forward 2 months later, has positive urine cultures. The urine cultures from 613 was again positive for Enterobacteriaceae, and now is more resistant. It has become resistant to Bactrim. And look what happened to ceftazine-avibactam. Now it has from 1 to 30 micrograms per mil and has become resistant. So the point that I'm going to make here is that these isolates become common and we select for them, then we may lose our ability to predict susceptibility with the molecular test. And we have to monitor these and make sure that we understand epidemiology so that when that does happen, we don't overpredict based on molecular testing. So this is my last slide. I want to thank all the people that have contributed to the work that I presented to you. The study went on for 4 years. So you can imagine multiple people were involved to be able to carry out such a long study. And so people in my laboratory, scientists in our clinical microlab laboratory, the bot group that did the whole genome sequencing and analysis, collaboration with Bruker and UC Davis to do the analysis, and collaboration with Fred Tenover at Cepheid. And our study was supported by Merck. So I'd like to thank all the individuals and organizations that contributed. Thank you very much.
Great. Thank you much. That was a super interesting presentation, especially like the part end, sort of a sense of what might be to come. You know, we had a number of questions that were sent via the Q&A function. Some were sent directly to the host, Lynette Corona. And I'll go ahead and just start asking a few in particular order. There was one question just about, you know, which hospitals, or was this a single hospital that was involved, and what breakpoints were used for a lot of these tests?
Yeah, so our hospital serves an adult pediatric hospital and their affiliated clinics, but I can tell you that majority of our cases were observed in adults, well over 90%. And we switched to the 2010 breakpoints, if I correctly, either in 2010 or 2011. So we are using the post-2010 breakpoints.
Okay, thank you. The next question was about, you know, you mentioned and you gave a great description of the non-carbapenemase or non-CP-CRE. And I was wondering, you know, for those of us in the audience involved directly in infection control, when you talk about the OMP-C and OMP-F, you know, how transmissible are these mechanisms of resistance? And are they a similar sort of threat that infection control as carbapenemase-producing CRE, such as KPC, NDM, et cetera?
Yeah, so the outer membrane proteins are part of the outer membrane of these organisms, and they are not transmissible on plasmids or elements. And we select for them to become mutated when we place patients on beta-lactams, essentially cephalosporins and carbapenems. So transmission, they're not transmissible. So for that reason, they're not as feared. And I also believe that they're not transmitted. The plasmid-encoded carbapenemase-producing CREs are transmitted. So you're not an infection control expert, but I know that they are not as much of a concern now. The question is, should they be, because the organism can be very resistant, right? They have a combination of phosphorinases and mutations that phenotypically make them look like CP-CRE. I don't know, honestly, whether there is comparisons to see whether they should not be feared as much.
Great. Our question is about, I hope I'm pronouncing this correctly, blasomycin. Blasomycin is approved today for CRE-related and complicated urinary tract infections. Do you know which carbapenemases does it target? And in general, are you able to comment on blasomycin at all?
I cannot. I don't have experience with it and know very little about it. Sorry.
wrap up the questions for you, Niaz. And again, thank you so much for this presentation. It was very interesting, and I think your findings really highlight the importance of carbapenemase mechanism testing and really to understand not only the epidemiology of these organisms, but also to help predict antimicrobial agents that might, or in many cases, might not be effective against certain carbapenemase producers. And especially this is the case in regions where, in your region, where there is such diversity of carbapenemases. And I'd just like to highlight that also, we're pleased that so many clinical laboratories are bringing the capability to do carbapenemase mechanism testing, but also there are several local public health laboratories that are able to do carbapenemase mechanism testing, and in addition to that, our state microbial diseases laboratory also offers this testing. So any microbiology laboratories out there, I think it's best to contact your local public health laboratory if you don't have capability to do carbapenemase mechanism testing in your laboratory, to explore what options might be available to you via public health. And that transitions us, I think, to our next presenter, who is Sandy Barla, who's going to present from the Los Angeles County Department of Public Health, their experience with carbapenemase mechanism testing in Los Angeles County. Lynette, able to tee that up?
I can do the same. In the left-hand side of your screen, you should see the Quick Start tab, and from that tab, you'll be able to share your desktop.
All right, myself a presenter first. Okay.
This is Sam at CDPH. I'll go ahead and mention, for those of you, I know there were some people that may have had questions that came at the end. Feel free to just, again, send them to Lynette or put them in the Q&A box. We'll make sure to address all of them by the conclusion of the webinar. Thank you.
Great.
Screen.
Slides. Thank you.
All right, beautiful. Thank you so much for giving me the opportunity to present on our CRE experience in LA County. Objectives of my talk today are going to be to describe the characteristics of the CRE that were tested by our public health lab in LA County, and then to also discuss their public health and clinical implications. I know Dr. Benai just reviewed this heavily, but I wanted to make sure for the purpose of my presentation that everybody understood the difference between carbapenemase-producing, or CP-CRE, and non-CP-CRE. The main difference here is that CP-CRE contain an enzyme that activate the antibiotics, thus making the bacteria resistant. And the enzymes are often encoded on a plasmid, as you can see here. The plasmids can be easily shared between different types of bacteria, kind of sharing a pizza with your friends. On the other hand, non-CP-CRE have other mechanisms that make them resistant, kind of like porin mutations or efflux pumps, as here, that actively pump antibiotics out of the bacteria. Within the carbapenemases, there are different classes, and again, this was already discussed. I won't go into it too much here, but I wanted to show that different classes of bacteria can be, classes of carbapenemases can be found in different types of bacteria, are endemic in different parts of the world, can be inhibited by different agents, and can, in addition to carbapenems, confer resistance to different types of antibiotics as well. The LA County CRE surveillance strategy consisted of reaching out to clinical laboratories to voluntarily submit the big 3 CRE isolates, which were E. coli, and Enterobacter species. The isolate sent to our public health lab, they also asked for the final laboratory result to be submitted, which included the AST results. We were able to enroll 33 clinical microbiology labs in LA County, which represent about 40% of acute care hospitals, 5% of long-term acute care hospitals, and also enrolled 1 large reference lab that serves 60% of the long-term care facilities here. The split was sent to our lab. The first thing we did was to do a MALDI-TOF mass spectrometry to confirm the organism's ID, and nanosphere to detect the carbapenemase genes. And Sphere is able to detect detect the 5 main carbapemases found around the world today. So between January 2015 to May of this year, our health lab received and tested over 1,000 CRE isolates. About 3/4 of these isolates came from acute care settings, and about 1/4 of these came from long-term care settings. And able to collect and analyze susceptibility data for about the first 661 isolates that were received from January 2015 to April 2017. And I'd note that the submitting labs did use different methodologies to test for susceptibility, but a large majority of them use an automated AST system. These types of specimens that were sent to our lab, half of them were urine isolates. We did obtain a mix of others. And as organisms go, we did obtain a majority were Klebsiella pneumoniae isolates. However, we did detect a couple of CR. coli and CR. Enterobacteriaceae species. Along with these, we did receive some other types of organisms, and these included Citrobacter, Morganella, and primarily Serratia as well. So now, getting into the genomic epidemiology, we've been finding that over 80% of our CRE produce a carbapenemase, which is significantly higher than CDC estimates of 34% and also higher than what was just presented before me. Almost all of our CP-CRE contained KPC. However, we did detect several NDM, VIM, and had our first confirmed IMP-producing CRE earlier this year. A few isolates had 2 genes identified, and it was usually KPC in addition to something else. This is about 15% of our CRE isolates that did not produce a carbapenemase, and we are unsure of the mechanisms, but we assume they are similar to what Dr. Benai presented on earlier. I'm just noticing the numbers might be a little bit off here, but wanted to show the trends of carbapenemases by the organisms that were sent to our lab. You can see here that it's primarily Klebsiella pneumoniae that is carrying these carbapenemase genes. However, E. coli and Enterobacter sometimes do contain these carbapenemase genes as well. And we will also take a look at the susceptibility data that was sent to us to look to see if there were any differences in susceptibilities between different types of carbapenemases. Now, the numbers there are not strong enough, but we did find the carbapenemases were not strong enough, but we did find that when looking between CP-CRE and non-CP-CRE, there were some differences. So first, for example, looking at the aminoglycosides, we saw that CP-CRE, highlighted here in purple, in comparison to non-CP-CRE in the next box, there were significant differences where the carbapenemase-producing CRE had lower susceptibilities to these adjunctive agents as compared to the non-CP-CRE. Looked at colistin, we found that the differences were not as significant. And looking at agents like tigecycline, we found that CP-CRE were actually more susceptible as compared to non-CP-CRE. And so this is speaking into the point that antibiotic activity can vary between CRE that produce a carbapenemase and those that do not. And one major limitation I'd like to point out here is that these data were provided by the submitting laboratory, which again use different methodologies for antimicrobial susceptibility testing. So some of these results, like colistin, might not be as reliable. However, we did use the most updated CLSAI breakpoints to calculate the percent susceptibilities presented here. And moving forward, our public health lab will be implementing susceptibility testing to confirm these susceptibilities and to also confirm the presence of any pan-resistant bacteria in our county. So in conclusion, our big finding was that a very large proportion of CRE in LA County produce a carbapenemase, most of which are KPC. However, non-KPC mechanisms are indeed present in LA County. And I also suggest that CP-CRE can be highly resistant to adjunctive agents for CRE. Our recommendations in light of these results are that we suggest public health laboratories to obtain access to genetic resistance mechanism testing in order to rapidly identify carbapenemase-producing CRE, not only for treatment implications, but also for the purposes of infection control. And important to communicate any CRE, especially CP-CRE, discovered in your facility to the next facility during patient transfer so that we can contain the regional spread of these mechanisms, because, as I mentioned, the plasmids that these carbapenemases are located on can be very easily shared, just like pizza. So it's important for us to work together to prevent this spread. To acknowledge and really praise our public health laboratory staff, led by Dr. Green, with Audrey Manolo and Julio Ramirez for their hard work in testing these isolates. And I'd also like to thank my HOU team members for their contributions. Any questions, you can ask me now, or you're always welcome to email me at the address here. Thank you all so much.
Andy, that was super interesting, and really to me some of the differences we're seeing in different parts of even the same state. So I think that's very, very helpful. Thank you. I wanted to read to everyone that if you have any questions, please put them in the Q&A chat, or you can send a private message directly to Lynette Corona, and she'll get them to us. I'll go ahead and start with a few. One of the first ones I discussed was just about the non-CP-CRE. It looks like you've found a pretty high proportion of CP-CRE, but for the ones that were non-CP-CRE, weren't one of those big five that you mentioned. Is there any phenotypic testing performed on those isolates, such as MSIM testing or carba-NP?
Those tests were not run, but moving forward, our lab actually just got its MSIM up and running, so we'll be doing that moving forward to kind of confirm activity.
Well, that'd be very interesting to see if there's, start identifying any kind of novel carbapenemases. In fact, speaking of novel carbapenemases, you mentioned that about 2.3% of the isolates had 2 or more carbapenemases identified. And in the past, here at CDPH, we've observed that patients that have multiple or these rare novel carbapenemases often have a history of travel or healthcare exposure outside the U.S. I was wondering if your team observed any pattern among patients who had a history of healthcare outside the U.S., or if you were even able to collect that kind of epi information about some of these.
Yeah, that's something we've been focusing on really here in LA County, is to chase down these novel MDROs, and that's through the non-KPC mechanisms. So, we weren't doing that at first, so I think I can't speak to some of the earlier non-KPCs that we found, as the records and the patients had already expired. But what we have been seeing is, I would say maybe 25 to 50% of them have had some exposure internationally within the year prior to coming to LA County. So I do believe that some of these novel ones are travel associated, and in fact, for those that had multiple genes, I do think all of them had some healthcare exposure internationally before they were detected here.
Thank you. And then I'll go ahead and ask sort of one of the more sort of questions that I'm sure you all get a lot, but it looks like there was KPC producing Klebsiella pneumoniae is the most common carbapenemase producing organism in LA. And do you have any thoughts, or your team have any thoughts on why this might be the case, and why in general there was a relatively high proportion, at least higher than what was seen in that CDC study, of carbapenemase producing organisms in California?
Yeah, we've been trying to figure that out for a while, and the best answer we can come up with is just that KPC is endemic. It's here to stay. So that's kind of the best answer I can provide. We're still kind of trying to assess the extent of KPC in long-term acute care settings, and hopefully learn a little bit more about how it's circulating and why there's so much here through future studies that we're doing.
What are your thoughts about the impact impact of carbapenem-resistant Acinetobacter and Pseudomonas, which are not Enterobacteriaceae, based on sort of phenotypic analysis, are concerning like CRE? Is there an analysis for plasmid carbapenemase carriage?
We have received some carbapenem-resistant Acinetobacters and Pseudomonas at our lab. We haven't been doing surveillance for them as of yet, so unknown. But for the few that have been submitted, we find that I think at least a couple of them did carry plasmid-encoded carbapenemases. So we actually just began receiving CRPA isolates at our public health lab and are going to be expanding to Acinetobacter sometime in the future as well. So we're hoping to get a better understanding of those patterns in LA County. However, I've been seeing in the literature that carbapenem-resistant Acinetobacter and carbapenem-resistant Pseudomonas may actually be more prevalent than CRE. So we're really excited and interested to see what those patterns are like here. But I'm not sure what you're seeing at the California level to kind of turn that question back on you.
Yeah, sure. And I was just about to make a comment. So this is Erin. At the state level, we also have been receiving carbapenem-resistant Pseudomonas as well as some isolates of carbapenem-resistant Acinetobacter. We do know that in general, approximately 20% of Pseudomonas is resistant to carbapenem Acinetobacter that change in the 50% level. So we are encouraging laboratories to submit carbapenem-resistant Pseudomonas or Acinetobacter. In particular, there are clusters or suspected transmission of these organisms. And when we have received those, we have found a handful of, in particular, Vim-producing Pseudomonas, including several instances where transmission seemed very, very likely. And with regard to Acinetobacter, similarly, we have identified at least one cluster extending across multiple facilities in California of a carbapenemase-producing Acinetobacter, and that was an OXA-237 isolate, producing isolate, and OXA-237 is actually quite rare among the carbapenemase-producing Acinetobacters. It does seem that these two organisms, although they do have prevalence of carbapenem resistance and lesser prevalence than the Enterobacteriaceae of carbapenemase production, carbapenemase producers among them do seem to be emerging in some instances and causing outbreaks and transmission. So again, we'd encourage submission of these isolates for carbapenemase testing, especially when there is a cluster or suspected outbreak of them.
And if I could just kind of, last thing, I was reminded by a colleague that actually our renal antibiogram data are showing that carbapenem resistance amongst Acinetobacter and Pseudomonas are significantly higher than carbapenem resistance amongst the Enterobacteriaceae. So I do think there's more concern for them here.
Right.
Did you find there was a disproportionate burden of CPCRE from specimens obtained in long-term care versus acute care? And I'll go ahead and add on there as a third category, long-term acute care, or LTAC hospitals.
I'll say that we kind of, for the purpose of this analysis, I kind of clumped the LTACs in with the acute care hospitals. So that breakdown I don't have on me, but I can definitely look into it more if you'd like. So, but we did see that there is higher prevalence of CPCRE in the acute care setting as compared to the long-term acute care setting. But even in the long-term care setting, we've been doing some point prevalence surveys in response to some of these novel MDRO investigations and in response to some outbreaks. And we've been seeing a bit higher percentage of KPC positive specimens than I think we had initially expected.
And, I know there's a number of people on the call today are probably from LA County. I was wondering, for my edification, did you, when you did all this data, over 1,000 isolates, have you noticed any large disparities within LA County, certain regions that or areas of the city counties that are higher prevalence than others?
Yes, the laboratories that are submitting to us are coming from all across the region, as far up as Lancaster and as far south as Long Beach. However, with this data, I haven't specifically looked to do for any regional patterns. However, when we were looking at our antibiogram data, some of our analyses showed that while there wasn't really any geographic variation, the rates can vary by facility and facility type.
Great. Well, coming up to the top of the hour, and we've, I think we have all the questions that I've seen in the chat box. I will mention, people about the presentation and slides and things like that. We will be posting some of these slides to our website, and we'll be sending out an email with a link for all the participants on the call today. It takes a little bit of time. We have to get it posted. There are some formatting issues, but we will go ahead and post some of these slides. We are not posting a recording of this webinar at this time. We will be sending out that information about the slides. I want to just send a big thank you to both of our presenters today. These were really fascinating. I think it was really nice to see them kind of contextualized with each other because about sort of similar topics, but obviously with different visions, and one from an acute care kind of perspective and a microbiology perspective, and the other from public health departments. So we're very thankful that you were all able to today. And if anyone has any additional questions that come up, you can feel free to email me. I think I've probably emailed everyone on this call many, many times. You can email our general box as well, and we can make those questions get answered. So I'd like to say thank you, and we will see you all next webinar, which will be in several months.
And email Sam if you'd like to get on the list.
Oh, yeah, yes, exactly. And I'll be sending out
webinars, yeah.
I'll be sending out a link for the list, as well as if the CLSI breakpoints is relevant to you or your clinical lab, please let us know. We'd be happy to work with them.
Yeah, tuned as well. Our next webinar, we're anticipating presenting similar findings to what Los Angeles County presented, but based on the experience at our state microbial disease laboratory with carbapenemase testing.
Great. All right, we'll see you. Good day, everyone.
Thank all of the presenters for today's informative presentation. This concludes our webinar. Have a pleasant afternoon.